Good afternoon, everyone. This is Michael Dolstein here, Chief Scientific Officer and Head of R&D at Pfizer. We have created really what I hope to be an intriguing panel. And as you may see, we have selected to symbolize that we are somewhere between normal and COVID in a hybrid status. And you can see three of our panelist colleagues on the stage, and three of us coming in through the video format. Jeff Spader, representing ICRIA as Chief Medical and Scientific Officer, is calling in from London. And John Houston, CEO of Arvinas, an intriguing biotech company, I think is calling in from Connecticut. And I'm here in Stockholm, Sweden, on some exciting conferences, but of course, I could find the time to come back at pre-Galien. I think for many of us, this may be one of the few and first times we are gathering hybrids or in the same room to discuss, celebrate, and point the way of where science is coming after the COVID years. This is a special occasion to speak about where will new drug therapies and vaccines come from, as we are celebrating the 50th anniversary of the pre-Galien movement and its role to serve innovation for humanity. Now, I think we'll have an opportunity to listen to the entire panel share thoughts about these topics. It is almost, I would say, a golden era of science translating into medicines and vaccines, although we come out of a tragic period for humanity with the COVID pandemic almost claimed now 5 million lives. The COVID pandemic itself became a testament to what science can accomplish under severe environmental pressures. We saw, of course, the very fast advance, 8 to 9 months, of two new technologies, of one new technology by two company groups, the mRNA vaccines from Pfizer-BioNTech and Moderna NIH. And I think it represented one example how a technology was incubated, maybe almost 2 decades. Among the major contributors were Drew Weissman and Kathleen Carrico, working at UPenn and other universities to evolve mRNA encapsulated in lipids to become a new platforms that company could further refine and apply to new use in the COVID pandemic. We also noticed during the pandemic time how quickly several companies responded in making monoclonal antibody against the spike protein, the very same target for the vaccines, and how quickly not only a single antibody, but a cocktail of antibodies were put together, proved late last year, and importance of the cocktails based on experience of how viral escape can quickly turn an effective therapy to making the virus resistant. A third modality beyond the mRNA vaccines, the mAbs, were small molecules. The first one in 2020, the first half of 2020, was the repurposing of remdesivir, belonging to the polymerase inhibitor class. Gilead were able to move its emergency use authorization for patients in a hospitalized setting. And more recently, we have seen other small molecules for oral use, while remdesivir was in an infusion setting. And One of them, Monopravir, was originally discovered by University of Emory, later moved into development by Ridgeback biotech investor group, and finally brought to positive phase 3 by Merck. Pfizer and other companies like Atea Roche have been working on protease inhibitor, also in record time for an oral drug to move into pivotal studies that are soon to read out. So in a way, what we learned here was a tremendous convergence of fast insight into high-resolution biology. From the initial cloning and sequencing of the SARS-CoV-2 by Chinese scientists, rather rapid structural determination, among them from University of Texas in Austin, of the spike protein by Jason McClellan. And all of those advances, of course, were instrumental to provide the biomedical insights, followed by the modality engineering, the platform engineering, whether mRNA, antibodies, small molecules, and the third piece of deep insight into large sets of data that were pouring into the medical journals and starting to build a momentum for understanding homologies between sequences involving functional properties of mutations and epidemiology impact as the vaccine was rising through unvaccinated, vaccinated, or infected patients. A remarkable year and a celebration what science can do in very difficult headwinds of a pandemic. But it was not just great advances that happened in COVID science. Several of the colleagues that are on this panel have this in the last year contributed with some unique data sets that brought new hope about how we can advance new drug therapies and vaccines for great needs among patients. Amgen got approval for a remarkable small molecule that can bind to a RAS-mutated oncogenic protein and show encouraging data in RAS-mutated G12C, famous mutation in non-small cell lung cancer, a breakthrough for patients with this particular precision indication. Amgen also generated data with another new modality, bifunctional antibodies targeting BCMA, that builds on many years of interest from Amgen in bifunctional antibodies, actually going back probably some 7 years with Blincyta from a collaboration with a German biotech. I hope you get a sense how new insights in science combined with engineering of modalities and unique collaborations has been part of many breakthroughs, whether for COVID or non-COVID. And I also wanted to mention from John Houston how his technology of Arvinas and the team that tailor small molecule to be dual functioning agents to bind and to allow degradation of proteins involved in cancer or neuroscience diseases. And John's team developed very intriguing data degrading the estrogen receptor with an oral drug. I think it's the first time that an oral drug was acting as a degrader, being a small molecule. And Michel Vonatzo's team at Biogen have in the last few years, including this year, advanced molecules that put its modality in a new setting. The aducanumab or Aduhelm, a monoclonal antibody for use likely inside the CNS, to deal with the amyloid burden of Alzheimer patients advanced through regulatory approval. And a number of years earlier, Biogen developed Spinraza or nusinersen together with Cold Spring Harbor Lab, Ionis, during a sequence of steps that became transformative for SMA patients, a genetic, very severe, fatal disease. So it's been an amazing period where the life science sector have been able to show what can be done and how diseases where there has been little hope or little knowledge quickly were brought into a maturity where they are within the reach for new therapeutic modalities. We'll also listen to Michelle McCurry. He's new CEO of the bioorganization, and look forward to a reflection from her and Jeffrey Spader from ICWA concerning how they see these new capabilities and the amazing speed that some of them have been able to pick up given extraordinary circumstances, whether we can see those experiences and capabilities continue way beyond these last years of remarkable progress, and what type of collaborative or urgency that has been brought to bear here do we need to embrace in the entire ecosystem to sustain this wave of innovation for difficult to serve diseases that at the same time open up new technology, letting in fresh air through the windows of suffering and turning undruggable space to druggable. Now, after that brief introduction, setting the scene, I will start with Michelle Vunatsos from Biogen. Your team has really pioneered the use of different types of biologicals, whether proteins or more nucleic acid-based, like the antisense Spinraza, and in an area that's been very demanding, neuroscience. And you remain a force in allowing learnings in neuroscience. So what's your view of where we are? Is this an inflection point in neuroscience? Are these new modalities able to change the difficulty we have seen in this area with this tremendous opportunity to transform care?
Thank you for the question. And we believe that the science is breaking in that space. We are dedicating the human talents and the bulk of our capital allocation to that space of high unmet medical need that is growing all around the world. And we understand that it's a specific space for which we don't understand always the physiology. Just imagine the pathophysiology. So it's high risk. And nevertheless, during the past 25 years or so, we have transformed the management of MS. We've been the pioneer for spinal muscular atrophy. And this year we've launched the first product that basically addresses a defined pathology of Alzheimer's disease with the clinical data well captured into the label. But nevertheless, we did face also failures. So it's a high-risk space, and we also communicated a few weeks back that we missed the primary endpoint for ALS, but with some encouraging data in the secondaries. So we decided to have an expanded access program so that we can collect more data and learn more. So we are pioneering in this space, even if we have 2 phase 3s in lupus and a biosimilars franchise. But neuroscience is the core of Biogen. And we believe that science is breaking, and with the support of technology, of omics, of basically addressing and selecting genetically validated targets, the biomarkers, and we can basically define homogeneous group into heterogeneous disease. So this gives us a lot of hope that we can find some subgroups where we can increase basically the probability of success for our clinical development. Nowadays, we have 26 programs in the clinic in neuroscience, and we have diversified in order to increase a bit the scope and have more shots on goals. And we are very active, as you know, in neurodegeneration. We are very active beyond Alzheimer's on Parkinson's disease, ALS, neuromuscular disease, but also major depressive disorders and neuropathic pain and the back of the eye. And we are really trying. We have nowadays 10 phase 3 or 5 products, 11, and science is progressing. We are encouraged by the fact that we can understand better the pathophysiology, and we can benefit of technology in order to understand better, and with the support of digitalization also coming that way, I think that we are in a better position to address this huge unmet medical need for which many companies were running away from, and again now with the regulatory approval of Aduhelm and some success that we've seen, we see other companies coming, and we welcome this investment in that space because epidemiology is mounting, and we need the support of everyone in order to basically provide some hope to this population in need. But obviously, technology and specialization is very important. It's not solely doing neuroscience as an aside. It's defining the specialty of the organization, the purpose of the organization in this vertical, from the people to capability. And this is the only way in order to potentially increase the probability of success. So this is where we stand, and proud to be making some progress in a very high-risk area, but for which the unmet need is absolutely tremendous. And I hope that for Alzheimer's, with Aduhelm, we are opening the door for hope. After 20 years not having any innovation in that space is absolutely terrible. Look at the progress for oncology or other disease areas during that 20 years, the way society has evolved, but for Alzheimer's disease.
Michel, your company built fame initially around immunology of both cancer with the rituximab contributions historically and its deployment in inflammatory diseases. And of course, you were the pioneers around multiple sclerosis, inflammatory or autoimmune disease. As you moved into other areas of neurology, such as Alzheimer's, it suddenly has turned into hypothesis that there is a considerable immunology inflammatory process in Alzheimer's, bringing your long experience from neuroimmunology to bear possibly in Alzheimer's. Do you think we should have optimism that this new clue to the neurodegenerative of Alzheimer's can be translated also into therapeutic intervention in the inflammatory process, not just in the neurodegenerative around amyloid and tau, etc.?
Absolutely, and this is the beauty of being concentrated into one space, is that you define interconnectivity between the programs. And at Biogen, we have defined basically trigger points and what a clinical outcome means for the other programs. And what you outline is potentially an area of hope, absolutely.
Thank you. Now to Bob from Amgen. The KRAS oral drug was technical and scientific dramatic advance. RAS and many different RAS mutations are considered a hard to drug key oncogenic trigger for many cancers. At the same time, your company also was advancing novel immunotherapies. What made you embark on such a tough and unprecedented area to use small molecules for mutant-specific oncogenes that were considered undruggable.
Yeah, well, thank you, Michael. This whole notion of what is undruggable is changing rapidly, and we're excited to have been able to advance Lumakras, which is the brand of the medicine that has been approved to treat patients who have a G12C mutation of their KRAS oncogene. And the impetus for that was a structural biology insight that made us believe we could occupy a particular space on the protein and interrupt the mutational effect of that G12C oncogene mutation. And we were obviously very aware that for 3 or 4 decades, scientists have been trying to do that. But I think it was a feeling that we had a differentiated insight into, again, the structural biology that enabled us to have the courage to invest as rapidly as we did to advance this medicine. And we knew that there were thousands of patients who had been told they have that form of mutation, for example, in non-small cell lung cancer, but also colorectal and other solid tumors. And we knew that they had been told there was nothing for them. So there was an urgency to our desire to identify the right molecule and get it into the clinic. And we were really excited, Michael. We were able to go from first dosing of a patient to having the molecule approved and launched in under 30 months, which for us was a record, and probably for oncology molecules a record, or certainly amongst the fastest that's been achieved. And the uptake for the molecule has been very favorable as clinicians and patients, again, react to having for the first time a targeted medicine specifically designed to treat the effects of their mutation. So it was an exciting moment for us as a company for sure, and hopefully also for the field, and helping to build confidence that what we have previously considered undruggable may be in fact druggable. And we certainly are devoted to trying to expand our understanding of what the druggable genome is today. I think if you read the literature, you might see about 20% of the genome is considered druggable today, and the remaining 80% was largely considered undruggable. But we think tools and technologies are changing how we ought to look at that. And so we're excited about continuing to identify novel, innovative, new pathways that we can advance for the benefit of patients like those that have the KRAS G12C mutation. So it's an exciting time, Michael. I think you did a wonderful job at the outset summarizing just how much has happened in a short period of time. And I think it's going to be very difficult for us all to keep pace with the progress, or even keep abreast of the progress that will be arising from our field over the course of the next couple of years. So one of the reasons we're investing as heavily as we are in human genetics and in the associated omics around the human genome is, again, to try to understand at the molecular level where there are diseases that we might be able to address utilizing the new platforms that are arising, and be able to go to patients across different disease areas and say, we think we have an innovation that may work for your specific disease. And I think we're on the cusp of being able to do that quite widely.
Your company has over quite some time being bold in acquiring platforms. I mentioned briefly you acquired a number of years ago Micromet for immuno-oncology, and initially with a very short-acting product that you now, I think, reported to can be extended into BCMA half-life extended product, and shown how you can bring new technology internally and refine them. I do recall also your move a number of years ago acquiring Decode in Iceland, which was at the time when a number of groups started to be hesitant that the promise of genetics in understanding disease will prove to be an easy path forward. Fast forward to today, genetics seems to be on the rise. The size of the databases, the improved phenotyping makes it likely a very valuable tool. We have seen the UK Biobank and many others dominating publications with eye-opening findings. How has the unique relationship you established with Decode helped you in the company? Is it primarily in discovery, or has it been transforming the thinking on possible precision medicine, early intervention throughout the entire company?
Thank you, Michael. Yeah, we're excited about the work that our subsidiary Decode Genetics does in the field of human genetics. We consider ourselves to be the world's largest company focused on harnessing insights, again, generated at the molecular level. Our conviction was not based on a belief that it would yield very rapid identification of new targets, but a belief that over time, the improved engineering in the sequencing field and in the ability to manipulate data with computing technologies would enable us to generate proprietary insights, and I think that is proving to be true. So something like 2/3 of our non-cancer portfolio today is address molecules that we think have genetic validation behind them. And so we're very excited about being able to use human data to determine where to invest in the discovery research field. So it's working very well for us, but it's not just limited to discovery research. It's also becoming a very important way for us to develop clinical trials. We like the idea, as Michel said earlier, of being able to go from homogeneous description of a disease to understanding the heterogeneous molecular nature of disease and try to customize our therapies precisely for those patients that will benefit most from them. So whether that's in molecules like Lp(a) or whether that's in our inflammatory portfolio, increasingly we see opportunities to use our genetic insights to help us decide where to do clinical experiments that we think the genetics tells us are likely to be successful. So it's an investment that we feel very excited about, a platform that we have built substantially since making that acquisition. And again, one that I hope over time will prove to have been a wise decision for us. You mentioned also our investment in Micromet, and maybe I can help set up your discussion with John by saying we think of, so our investment there is in a platform of bispecific T-cell engaging molecules. And our first molecule that harnesses that technology is known as Blincyto. It was the first ever bispecific approved for cancer. It's a molecule for which we've generated overall survival, favorable overall survival data for patients suffering from acute lymphoblastic leukemia. And it's directed against the CD19 antigen on the tumor cells, and it binds CD3 on the T cell. And the effect of binding those two molecules together enables us to redirect T cell lysis to the tumor cells. So this is a platform that we think of under the umbrella term of proximity biology. And increasingly, we think proximity biology, or the idea of having inducible drugs that enable us to induce a biologic effect, so we also call it the inducible proximity platform, will enable us to take a lot of targets that were considered undruggable and render them druggable. So in the area of targeted protein degradation, for example, we have a very large effort now, but our focus there is beyond just targeted protein degradation. We're looking at other things that we think we can achieve by pulling together molecules in either an obligate or non-obligate way to create an effect that's never been seen before in biology. But so it really is an exciting time, and I think both Decode and our commitment to bispecifics will play out over the coming decade.
Thank you. That was a great setup, how you pioneered proximity in biology by large molecules. And John, CEO of Arvinas, I think you have brought new, fresh innovation into the small molecule space, which lost some ground to all these fancy genetic and biological platform, but now being refreshed with the proximity biology induced by small molecules. And maybe you can expand what brought you into this space and where you see it go.
Yeah, thank you, and great to be on the panel. Yeah, Arvinas was founded in 2013 on the back of some great pioneering work by Professor Craig Crews and Ray Deshays as well. They were colleagues. And the first PROTAC paper was 20 years ago. Both of them are on that paper. But in 2013, Craig formed Arvinas as a spin-out of Yale. And his focus was, can I create a small molecule that can hijack the cell's normal protein degradation process. In other words, recruit a specific E3 ligase and be able to show that by binding to a protein that you wanted to degrade, you could bring these two proteins together into close proximity, and ubiquitin would be transferred from the ligase to the target protein, and that would serve as the natural signal for the protein to be dragged off to the proteasome, which is the cell's natural garbage disposal unit. So the protein's dragged there, shredded down to peptides, and flushed back into the cell. Well, those initial concepts were done in 2013, and he had created a really nice small molecule chemistry team. And they built up this set of rules over the first few years of Revinis, trying to understand how that technology could work. And roll the clock forward now a few years, and we now have a number of different breakthroughs that have occurred in the technology. We're able to show preclinically that in settings where you have resistance, overexpression, gene overexpression, aggregation, and even scaffolding function proteins, that a degrader can work very effectively compared to an inhibitor. And the most striking examples of that are our two lead programs in the clinic, ARV-471, which is an estrogen receptor degrader, which is now in phase 2, which has what we believe to be, at this stage of development, a best-in-class profile and showing really differential biology as a targeted protein degrader, and ARV-110, which is an androgen receptor degrader, which equally is showing a very, very good profile in a very late-line patient population. Now, we believe that these proof of concepts, and both in terms of showing that a small molecule, heterobifunctional molecule that's binding to two different proteins and inducing proximity, can act like a normal small molecule. You can see good PK, good PD, and now we're seeing clinical efficacy. So it's a very exciting stage for the technology. And we see that by the plethora of different companies in the space now. We were in splendid isolation for a few years, and now we're surrounded by 30 to 40 other small molecule based degrader companies, which is fantastic. And we think there's at least 10 that we know of compounds in the clinic that are degrader compounds. So the next few years, I think, will really show the utility of this new modality and show that it can actually get into the undruggable space in a significant way and drive differential biology in a way that we haven't seen with inhibitors.
Thank you very much. And I know, as we spoke about neuroscience earlier with Michelle from Biogen, that you are not hesitating to think about moving degraders into the brain. You have disclosed that you're building a pipeline here. So do you see opportunity to find new biology with this approach that hasn't been accessible before with other modalities?
Yeah, when I joined the company in 2017, they had great proof of concept preclinically on a set of oncology targets, two that I mentioned and several others. And we now have quite a significant oncology portfolio. But intriguingly for me, they had also tried an experiment where they designed a PROTAC that could degrade tau. And the way they showed proof of concept by it was by injecting the PROTAC into the brains of mice and showing that, sure enough, the tau was degraded. So I thought this was very exciting data, but the challenge was, that's great, but we're not going to be injecting this drug into people's brains. So let's see if we can make the PROTAC brain penetrant. So for the next few years, the team worked on that. And as we did that, we built out a neuroscience team. And we now have a significant number of brain penetrant PROTACs. We've worked out how to do that. And we now have, well, we've disclosed at least 4 of our targets. We have several more. We're working in neurodegeneration. And the next challenge for us is getting into the clinical candidate stage and moving this pipeline on in the same kind of way as we have with oncology. And of course, with PROTACs, the disease areas are not limiting. We chose oncology. Neuroscience was another big challenge, and we decided to deploy it there because we do think we've got a chance to get a small molecule approach to drugging these targets in a very different, unique way that's not been done before. But there's other disease areas that this technology could be adapted to. And we even created a joint venture in the agrichem space with Bayer. We created a company called Earth Bio that's using our technology to look at plant health and disease control in that setting. So this is an adaptable technology platform that's really showing its paces at this early stage of development, both in human disease and in agrichem.
Thank you so much. Now to Michelle McMurray-Heath of Bio. I think we have heard some remarkable efforts, how companies have been bold and courageous in bringing new technology to bear for unmet needs. And while the COVID put a barrier to the traditional way of working, many of the colleagues you're listening to were still able to reconfigure themselves, reinvent themselves, and continue to push forward in an unprecedented manner. So, Michelle, what do you think is important for rewarding that dramatic progress? Do you think we will see federal agency regulators rising up to this promise and finding ways to expand the use of breakthrough approaches, breakthrough designation? What are the concerns, whether in policies or in the political system, or the opportunities to maintain or slow down this quite impressive progress?
Well, thank you, Michael, and thanks for the invitation, and thanks for all of the folks who have waited through the entire day of this great symposium. This is a very tenuous time. We've seen such amazing progress in the biopharma industry over the last 18 months, and yet we have never been more disparaged or despised or distrusted at the public official level. And we really need to think about how we find better ways to tell our stories. So if you think about what happened during COVID, and Michael, you touched upon some of the highlights, but basically during COVID, our companies started over 1,000 research and development programs within the first 13 months. We're looking at a probable success rate when it's all said and done of 1 in 10, which is what we have seen so far in the rest of the industry and the rest of the disease areas in which we do research. Those kind of odds are incredibly challenging, and you have to have optimism to be in the biopharma space. So we need to find ways to take our policymakers on that journey to see that this is, while we deliver amazing miracle cures in many ways, there is a very rough path to get there. And there are a lot of ideas that fall by the wayside on the way. This would not be possible without policy adaptations like breakthrough designation. And this is not the time for us as an ecosystem to lose any fervor in the importance of designations like breakthrough therapies, because it's so essential that when we are moving into an unknown disease area or a rapidly evolving knowledge area, that we are not waiting on long preconceived clinical outcomes, but we are using what we know about the science and the biology to be able to detect successful compounds and biologics sooner rather than later, because the patients are waiting for them. And yes, it might not lead to the perfect drug first, but it leads to hope and progress and more data that allows you to continue to iterate and evolve the space. We have breakthrough designation because of HIV. The very first drug through it was AZT. Was that a perfect drug? No. But look where we are today with our ability to treat in that space. And it's all because we had the ability to start the engine and then follow the progress. And we have to do more and more of that. But it's going to take scientists and business leaders in this space to really stand up and be bold about telling the impact that you potentially have on patients and that you've already had on patients, because that's really what matters.
Thank you. Michelle, during the pandemic, advancement of potentially breakthrough treatments for a severe need that was so rapidly overwhelming the medical care system, like SARS-CoV-2, was facilitated by emergency use authorization. And while not a perfect system, it clearly brought medicines and vaccines much faster to patients. Most of them likely remaining to be extremely important contributions, and a few that were very early approved turned out to be less of impactful. Do you think it requires another pandemic for emergency use authorization, or do you see existing epidemics of many diseases that are not just induced by infections, but by environment in general? Do you think similar ways to rapidly give patients hope can be deployed, or do you think there is still the conformity of regulatory agency to follow the existing pathways and step back from those extraordinary efforts performed by FDA, but maybe not using them again for similar rapid service to patients? What's your thinking, and how do we make the best learned in this pandemic become a new part of the future?
Well, emergency use authorization is a very specific pathway that's really targeted at, a crisis, an immediate crisis in public health. And unfortunately, we have many more long simmering chronic disease crises that we don't really notice because we're the frog in the pot that's getting boiled, right? But we have lots of regulatory pathways at our disposal today between accelerated approval, breakthrough designation. There's a lot of ways to say, for regulators to step in and say, this is an important advance. We're going to get it out to patients who are waiting, and we're going to continue to collect more data as we go. That's what we have to double down on and reinforce, because it's so important for all the progress that we make. The other thing we need to be very careful that we do is we need to completely renovate our clinical trial system. So one of our member CEOs at BIO, we represent over 1,000 biotech companies, small to large. One of our CEOs likes to say, I spend 90% of my time and 90% of my cost on the clinical development portion. That's obscene. It's obscene because often we're chasing endpoints that are nice to know rather than need to know. It's obscene because we're letting the perfect be the enemy of the good almost each and every time. And it's not just a question of forcing company A or company B to do more and more work to go the extra mile. It also has a huge impact on patients that are participating in clinical trials when they don't need to be, and patients who are waiting on those final approvals that could have come sooner. So we need to stand up and demand more use of real-world evidence. We need to have a very dynamic regulatory system that says, after a drug goes on the market or a biologic goes on the market, we are going to continue to monitor in real time, which we should always be doing anyway. So if any new safety signal or any new knowledge is learned, we can course correct. We don't want regulatory systems and decisions that are set in stone. We want our regulators to be dynamic and to adapt with the science. And that's not what we're encouraging right now. So we need to do a lot more of that. And then finally, it's time for us to ask ourselves, is placebo-controlled double-blind trial really the gold standard? We hold ourselves, even as researchers, put regulators aside, to that gold standard as the optimum. That's what we're really looking for. And yet, most people know, or definitely our regulators know, that what we get out of those randomized controlled trials are never what we see when the drug or biologic is actually released to the general public. It's because our trials are not diverse, it's because our trials are artificial, it's because our trials exclude so many other comorbidities. And so we have these pristine bodies of data that mean nothing in actual medical clinical practice. And so we need to say, there's a faster, better, cheaper way to start getting this data, which is more conservative of human life and human suffering. And that, I think, is the path forward for us as an industry.
Thank you so much. So Jeff, what's your thoughts and guidance about new capabilities emerging and the various phases of R&D? Where do you see the biggest opportunity for progress?
Sure, thanks, Mikhail. I think I want to just touch on something that Michelle just talked about is the patient, right? When we look at the what has really advanced during the COVID outbreak, I mean, we see amazing science. We see a very adaptable regulatory pathway, but we also saw patients and people willing to participate in studies. If we didn't have them in these studies, we wouldn't be able to identify whether it's a placebo control or some other type of way. We would not have been able to have conducted these studies and gotten the insights that we need. So I think one of the great takeaways is the engagement of patients and how do we make it easier for patients to get into studies. We know that less than 2 or 3% of eligible patients participate in clinical studies. We know that half of principal investigators who are new principal investigators, half of them will never do another clinical study again. The way we have the traditional clinical development program is a burden to patients, it's a burden to principal investigators, and it dissuades people from wanting to participate or make it difficult for them to get into studies. So I think one of the great outcomes of the silver linings of a terrible COVID crisis is the ability to reach out to patients more effectively, more diversely, to go to where they are. And I think we see some of the hybrid types of virtual studies combining the bricks and mortar with a virtual type of setting that allows people to come into studies more readily. And frankly, the transparency that we saw with especially the COVID vaccines, the publication of protocols, that gives credibility to patients for them to enroll into studies. And I think anything that we can do to make it easier for patients to come into studies, make it easier to go into medical records, to be able to match patients appropriately, anything that we can do that reduces the burden on investigators from a regulatory perspective and make them more willing to participate in research. I think those are all great things. I think the other thing I would just point out is the use of real-world medicine or real-world evidence. We literally saw insights coming out almost on a daily basis in places like Israel, which had a great centralized medical record system, was able to provide some insights, again, maybe not perfect, maybe with caveats and with limitations, but it allowed us to get insights, kind of what Michelle was talking about. There were other ways to get insights that were independent of a placebo-controlled study. I hope that's one of the things we take away from this pandemic.
And if I may add, Mike, to what was just said, I think the use of digital biomarkers can be extremely useful. We just initiated with the collaboration of Apple a study with, if I'm not mistaken, 23,000 patients using basically iPhone and all the cognitive functions that the use of iPhone can detect, so that we can eventually move towards the left in terms of early detection of defect for patients who potentially are at risk to develop Alzheimer's disease. So whenever we can quantify, use biomarkers and somehow eliminate the sometimes subjective judgment of a clinician for measuring a clinical improvement might be important.
That was a great comment. As you spoke about biomarkers, there's been a lot of enthusiasm in many companies, including Apple, for the use of artificial intelligence to better tailor the right product to the client. Do you foresee, Michelle, that the use of artificial intelligence can be applied in the relatively near future to tailor the right drug for use in a proper patient that have a relevant prognosis?
I fundamentally believe that the use of machine learning, AI, will accelerate clinical development, will increase the probability of success. At Biogen, we're investing heavily in that space for all the entire value chain of the company, from early research to engagement with the customers. We have a team of 200 highly qualified individuals and partnerships. We have collected data for example, within MS, for more than 25,000 patients for many years. So well-organized data from blood to CT scans and other set of data, clinical analysis and assessment. And out of good database and data analytics, we can eventually query and build data intelligence that could eventually could revolutionize the understanding of the disease. For example, is it a neuroimmunology reaction in the case of MS, or is it a central disease where we can see eventually, based on the imaging, a defect or a signal prior to the immune response? Well, if you have data, at least you can ask yourself the question. So it's fascinating the type of things we can do with technology, and we are very encouraged by the potential benefit in that space, certainly.
Thank you very much. So let's see if the organizers are able to get comments from the floor or remote participants.
Would you go to the microphone? Yeah.
Mrna Vaccines, I actually consulted most top pharma, Pfizer, Merck, Novartis. I've been in commercialization and clinical trials prior to that, and then alliance management. I'm just wondering that in terms of the paradigm now, in terms of collaboration, is there ways that, pharma is starting to look at collaboration across the board? I know I mentioned a question before about, the trial design is kind of old, schematic in the past. We need to look at it diversity. We need to look at it from a global perspective as well. But on an added sense, I mean, with Alzheimer's, with MS, there's a lot of startups that and I've been doing this as well, assisting venture capitalists as well as startups, marrying them together. There's a lot of startups that are unique, that are innovative. How does big pharma, bring them in collaboration-wise, other than accelerators or the venture arms of these pharma companies these days?
So
maybe
I believe
Brad Wave.
Okay, go ahead.
Could I respond from
Sure.
Galien here?
And Michelle, please amplify with your thoughts as well. We're engaged every day in discussions with startup companies and collaborate with them across a variety of different, or using a variety of different means. Sometimes it's specific investments in programs. Sometimes it's equity investments as well as project-related funding. So I think that's something that the industry does very well. I think the growth in the number of innovative companies, as Michelle said, there are already 1,000 entities represented inside her organization. I think so the growth in those entities gives us an opportunity to do more and more of it. But as an industry, I think we've done it well, and I think you'll continue to see a lot of that.
So I believe it's inconceivable, even if we are highly specialized into one space, that we believe that we can master, all the science going on. By definition, there is much more going outside of our words. In the room, we have Dr. Gallagher, whose mission is excellent innovation and partnership very early, so that we can pool capabilities, knowledge from other parties, including other pharma companies. Because we are in the same boat, and each one is learning from each other. So absolutely. And there are some very early research investment being done on the genome based on large population base, where we pool the capability and investment from different pharmas.
Thank you. Any further question from the floor or the online community?
I'll just add to what Bob and Michelle just hinted at as well. We are seeing an explosion of collaborations. We set up a Craigslist type hub for companies to be able to trade resources, advice, supplies during COVID, and it was completely successful and well utilized. In the last 10 months of this year, we've seen 44,000 partnering meetings occurring on our platform. Those are small biotechs talking with large pharma, talking with investors. So there's an explosion of hunger to partner, to collaborate, to work together. And I think that's the biggest success story of our industry, is that we do that very well, and we just improve on it each and every year.
Thank you.
Mike, maybe here in closing, I can just offer from the rest of us to you a congratulations and a thank you for the incredible work of Pfizer and BioNTech over the course of the past 2 years. We're all benefiting from the speed at which you in particular mobilized your organization for all of our benefits. So thank you for that, and I hope everybody's inspired by the incredible innovation and speed and scale at which Pfizer, BioNTech were able to move, and not just them, but clearly an exemplar of what we hope this industry can do now post-pandemic as well. So thank you, Michael. And it looks like we had a chance to answer everybody's questions in the room. I don't know if there are any online, but if not, maybe we say thanks to the audience here. And
I thought, Bob, we could just close with a quick walking around the table. I think there is still some time here, right? Or it's hard sitting in
it's
Europe
11 o'clock in Sweden, Michael.
Yeah.
It's late in Sweden.
A quick, let's get one sentence from you all. If we were here October 28, 2026, 5 years from now, what would be the key topic, the question you would like to be asked, or the progress you would like to report, October 28, 2026? Michelle from Bio, start. And just a one-sentence punchline so we can go to bed soon in Europe.
So my boldest dream will be that what we consider today as intractable disease become chronic, manageable disease. The way Michel described, HIV was a death penalty in the 80s, and today it's very, very well managed as a chronic disease and people live a normal life. This is what we should aim for the ALS, AD, Parkinson diseases of this world, for which the mortality is just tremendous.
Thank you. Michel from Bio, what would a typical company that is a member in your organization on that day, 2026, what type of progress do you think will have happened?
That we've repaired the mistrust and created trust with all communities that we serve, because patients see us in the research we conduct and in the products we produce.
Thank you very much. Jeff Spader.
Yeah, I think I'd echo that we'd like to be able to see patients being able to participate in clinical research and feel comfortable doing it, and feel safe in doing it and want to do it, and seeking it out on a regular basis. And I think it may be in terms of a therapeutic area, mental health and behavioral health. I think we're at the very beginnings of a paradigm shift there. I'd love to see where we are in 5 years in that space.
That was a really great encouragement. Mental health, psychiatry is one of the areas that really need collaborations. I've been very pleased to see, I have co-shared for many years with Francis Collins an industry consortium, Accelerating Medicine Partnership. Many of your colleagues are there. And more recently, finally, after 7 years, the first collaborative effort for a psychiatric disease emerged. And I really felt proud for the medical community as a whole to come together and create a new knowledge bank. So I really echo Jeff. John Houston from Arvinas. So will they still be called PROTAC, or would there be a new acronym celebrating further ingenuity in this type of multifunctional small molecules?
Yeah, that the question I'd love to be asked then in the future would be, what was the world like before protein degraders? Because that would tell me that we've made a big success of it, not just Arvinas, but all the companies involved in it, both in oncology and in neuro and all the other disease areas. But yeah, I'm hoping the next few years prove that this is another modality that can be added in to all the modalities that companies use right now.
Okay. Bob, thank you for the kind recognition of hard work by us, all our collaborators and patients. I hope you all feel safe being vaccinated, whether you had 2 or 3 doses. We can discuss another time. But what would be the key topic Amgen may be celebrating that late October 2026?
Well, I hope in 2026, somebody asks us what we meant in 2021 when we said undruggable, because like John, I'm hopeful that some of these new technology platforms render that question irrelevant.
Thank you very much. I hope you all in the room and online participant got some encouragement of the rapid pace of progress that we have seen and opportunity ahead of us. Continuous collaboration, transparency, courage to take on big questions is what we would like to see, and that the ecosystem from regulators, academia, venture capital work together with large or small companies to advance medicines. And like Michelle from Bio said, hopefully we can also develop a deeper relationship with the public across many regions of the world on how much we can really change when we have a new way of working together and not fall back into the past of being incremental. I wanted to thank the great panelists for sharing their views, telling us insights from their companies. I hope you will enjoy the upcoming reception and ceremony at Galien. I feel really sorry to be missing it, but I look forward to read the news headlines. And I took note that the Roy Vagelos Pro Bono Humanum Award for Global Health Equity will this year goes to CEOs of 6 companies: Moderna, J&J, Pfizer, AstraZeneca, Regeneron, and Eli Lilly, behind the vaccines and therapeutics in front of the response to COVID-19. But I hope that you also heard from all these colleagues here, so many other miraculous advances of science happened during this difficult period. So thank you all. Good night from Europe. Good or late afternoon in US.
Thank you.
Thank you.